Hello, my name is Margit Tolle, and the title of my today's lecture is The Influence of Effective Microorganisms on the Growth and Nitrate Content of Vegetable Transplants. Introduction. Effective microorganism technology was first developed in the 1970s. EM is a mixture containing primarily lactic acid bacteria, photosynthetic bacteria, and yeast. Maintain that pH 3.5. EM suppress the incidence of pests and diseases, solubilize minerals, conserve energy, increase photosynthetic efficiency, and fix biological nitrogen. Problem: elongated low-quality vegetable transplants. EM. The purpose of this investigation was to assess the influence of effective microorganisms on the growth and nitrate content of vegetable transplants. Materials and methods. Experimental site and time: Estonian Crop Research Institute greenhouses, winter of 2014. Crops: cucumber, pumpkin, squash. Substrate: peat-based mixture, fertilized with peat care, 11-25-24. Magnesium sulfate and mixed with dolomite lime. Materials and methods are continuing. Treatments: EM treatment and control treatment. Seeds were soaking half an hour in EM 1 to 500 solution and in control treatment in water. Substrate treatment with EM 1 to 500 solution and in control treatment with water. 3 true leaf stage spraying with EM 1 to 1000 solution and in control treatment with water. And 1 week later, the same solutions, again 1 week later, the same solutions, and again 1 week later, the same solutions. So in total with 4 spraying with EM solution or water. Materials and methods are continuing. Replications and experimental design. Randomized block design, plot size 6 plants, 4 replications, experiment repeated 2 times, laboratory analysis, nitrate content, statistics, ANOVA by Excel 2010, comparison by means by Fisher's LSD test. Results: the head of cucumber transplants treated with effective microorganisms. You can see that the rate of cucumber transplants was lower in EM treatments than in control treatment. The stem diameter of cucumber transplants treated with EM. You can see that stem diameter was greater in EM treatment compared to control treatment. The content of nitrates in raw cucumber transplants. You can see that the content of nitrates in raw cucumber transplants in EM treatment was lower than in control. The height of pumpkin transplants treated with effective microorganisms. You can see that the height of pumpkin transplants was lower in EM treatments than in control treatment. The stem diameter of pumpkin transplants treated with effective microorganisms. You can see that the stem diameter was greater in EM treatment compared to control treatment. The content of nitrates in raw pumpkin transplants was not statistically different. The height of squash transplant treated with effective microorganisms. You can see that the height of squash transplants was lower in EM treatment compared to control treatment. The stem diameter of squash transplant treated with effective microorganisms. You can see that the stem diameter was increased in EM treatment compared to control treatment. the content of nitrates in raw squash transplants. EM treatment had lower content of nitrates in squash than in control treatment. Photo of squash plant on the left, EM treated plant on the right, control plant. You can see that the stem diameter was greater in EM treated treatment. Photo of pumpkin transplants on the left, EM treated plants on the right, control plants, and again the stem diameter is greater in EM treatment. Photo of cucumber plant on the left, EM treated plant on the right, control plant, and you can see that the EM treated plant is lower than a control plant. Conclusions: cucumber, pumpkin, and squash transplants were short and stem diameter was greater in effective microorganism treatment compared to control. Cucumber, pumpkin, and squash transplants contained less nitrates and effective microorganism treatment than in control treatment. Acknowledgements: the present research was carried through with financial support from Estonian Agricultural Registers and Information Board and with the help of the Jaagumäe Agro OÜ and Estonian Crop Research Institute. Thank you very much for listening.